Live streaming is on.
Okay. Yeah. Eric, there is a microphone that does not work inside the room on the table. Yeah. Is the live streaming on as well? Nicole, can you?
Yeah.
We are trying to solve a problem with the phone on site, and then we are ready.
Because you had the internet. Yeah, it's working now.
Yes. Now they.
This
yeah,
one,
now
no.
they work.
This one is out of order. Oh shit. Yeah, water we should not have.
Temporary. This one.
Yeah, but we can continue with 3 and then we solve it later.
Yeah. Yeah. As long as. So this works. As long as 3 work, we're good. Found it. This was disconnected. This one was disconnected. Yeah. I connected it myself.
Pardon?
But it's okay. Yeah, it's connected to itself. I see. So it should be connected. Turn this off again.
Still 5 minutes. Or 2.
You can work with the microphone or not?
We are almost connected.
think it's working. Yes. Yes. So now we are all set up. No. Microphone. No. Now it's better. There is an echo somewhere. Somebody has a. It's good? Okay, thank you. So, good afternoon, evening. No, there is still a problem. Let us check again. Where is it? The volume. No, it's off here. Does anyone have the volume? 1 2 1 2 1 2 2. It's okay. Seems okay now. Good. Let's start the session 9 on water-based or wastewater-based epidemiology and risk assessment. We'll have a nice keynote of Colleen Nocton. I will let Frances chair the session. I would like first to warmly thank Colleen to be with us. We know each other from the University of South Florida. And Colleen, besides being an assistant professor in Merced, she's also an Iron woman, so a specialist of triathlon, and she will speak about COVID-19. So please, Frances, take the floor and enjoy this chairing.
So the first session, the keynote, the keynote speaker session. What happens? Hello, hola, 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 hola. Okay, again. The keynote speaker of the last session of MIWI is Colleen Nocton. She's an assistant professor in civil engineering and environmental engineering at the University of California, Merced, and has more than 10 years of experience working in Africa, North and West, and focuses on global water management, sanitation, hygiene, climate, and food security. And during the last couple of years, she's been working in the COVID Poops 19 initiative, which has drawn a lot of attention internationally. And the title of the speaker of the session is COVID Poops 19 and Beyond: How the Pandemic Has Changed the Water Sector.
Good morning or good afternoon, everyone. Thank you for the wonderful introduction. I'll share screen now. Told I need to. Okay, hopefully everybody can see that. Let me know otherwise. Sorry?
It works very well.
Okay. Yeah, again, thank you for that warm introduction, and thank you again for this opportunity to be a keynote speaker at IWA MIWI. It's a great honor, and thank you for providing opportunities to early career researchers and being mindful of the diversity, geographic and ethnic of your speakers. And it's been a very well organized conference, and I know it's very challenging to do a hybrid and virtual conference. It's like 2 conferences in one, and this is the last session of a marathon. So thank you for all those joining right now. It's been a very intense conference. I've been catching up afterwards, given the time zone, and I've really enjoyed and learned a lot so far. So as you said, I would present about COVID Poops 19 and beyond, how the pandemic has changed the water sector. So I'm the principal investigator behind the global dashboard for wastewater monitoring of SARS-CoV-2, if you haven't seen it yet. And though I'm presenting this work, it's on behalf of a lot of collaborators, students, postdocs, and other professors, especially for the Global Data Center, Dr. Joan Rose at Michigan State University, Dr. Gretchen Medema. So I would like to thank them, and I'll acknowledge them at the end of the presentation as well. I'd also take a moment to acknowledge the indigenous people of which the land that UC Merced is located, the Miwok and Yokut tribes that we continue to learn, live, and work on their traditional homelands. I am using subtitles. They're not perfect. We do use them here at UC Merced sometimes for hearing impaired, and also since I'm more soft spoken. So hopefully they're not too distracting or help you if you're listening now. And so today I'll present about a background about wastewater-based epidemiology. I know this conference or group is probably very informed about WBE. Just a brief background as we lead into the dashboard and our methods behind it. And then we've been privileged to be part of a global data center, WSphere, and talk a little bit about WBE beyond COVID-19. I'm really looking forward to all the speakers in this session, some even talking about Zika and dengue and other pathogens trapped in the wastewater. It was great to see all the posters. I think there was like more than a half dozen on wastewater-based epidemiology, and talk a little bit about how I feel and have reflected in this past year how the pandemic has changed the water sector, and then welcome any questions or comments in the chat. So wastewater-based epidemiology, environmental surveillance, wastewater surveillance, wastewater monitoring, however you want to term it. I think wastewater-based epi is a little bit more recent term, but just for tracking pathogens in the wastewater for public health use. So it's really become prominent in the pandemic and COVID-19 that we've seen that people also shed SARS-CoV-2 in their feces, about 40 to 80%. Depending on the papers, we're still estimating that. Though we found it's a primarily aerosol-based respiratory sort of spread human to human. There is shedding in the wastewater that you can detect, and this can give you a larger sample of the population. Wastewater treatment plants can serve millions of people, or you can monitor in the sewer shed or building level, or smaller treatment plants, hundreds of people, so you get a more representative sample of the population, and it can detect both asymptomatic and symptomatic cases. People are shedding even when they're asymptomatic or before they're showing symptoms and spreading in the population. So this has been shown to be a potential early warning indicator before you see it in the clinical testing data, or especially if you don't have robust clinical testing. But WBE isn't new. It's been used for decades. And many of you probably at this conference have been working on it for a while. You do see it in the media or news stories on television, them talking about a new or novel method. So it's been quite new for the public, but it was used in poliovirus and actually tracking vaccination rates there, as well as like hepatitis, adenovirus, norovirus, lots of different pathogens before and continuing moving forward. So WBE, like some of the first papers for SARS-CoV-2 came out of like the Netherlands, Dr. Medema, and then Awarish Ahmed in Australia. And we also saw like the first, cluster of cases where it really strained the healthcare system outside of China and Italy. They started using it early in the pandemic, but I think papers were coming out like in March 2020 as people oriented the methods for SARS-CoV-2. So as I said, there's a potential early warning indicator. The recent literature is showing more like 4 or 5 days, anywhere from 2 to 7. Sometimes it more follows the clinical data. If you have a very robust testing campaign, it's just another check, and kind of it helps you. Public health departments still like to use it to show the community spread, and it can be really useful. And so even at the building level, a lot of universities, I think we have 265 now in the dashboard. I did a recent update, and they were using it to test at the dorm or building level. And if you start getting a positive signal, then you know to test the rest of the like 500, 300 so students. And so they were even able to estimate that they thought there were 2 cases, and they tested and they found the 2 cases, so they could isolate those students before it became a wider outbreak, before we kind of had the vaccination mandates as well. And it's still being continued to be used even with vaccination, since there's still some spread. And this is just an example of some cities, and Ottawa was really prominent, as well as many cities throughout the world, in Europe and the US and in South America. So many of you may have heard of the Johns Hopkins University dashboard that was really helpful for clinical data and tracking the spread of the pandemic and unfortunately the deaths, but fortunately now the vaccination rates. And I was like learning about WB for COVID. I had heard about it before, of course, but seeing really on Twitter it exploding, like lots of people monitoring and like stepping up in the pandemic to help out. And I was like, oh, it would be great to have like an early warning dashboard with the wastewater data. And I didn't really see people aggregating this information. So I applied for a COVID-19 seed grant through the Center of Information Technology Research in the Interest of Society. It's like a center for 5 different University of California campuses, and was able to get some funding for some students to work and track this information. Of course, it built off of a lot of efforts that were happening globally. One was the Wastewater-Based Epidemiology Collaborative run by Kyle Bibby's group and with Aaron Bivens. And then there's the Research Coordination Network. Of course, I know a lot about the US efforts. I do keep track of like Europe and Asia and other places as well. But we really they were starting to collect all the publications that were coming out, and they had a Slack channel. We attended webinars at all hours of the day. I felt bad my students were going to the Asian webinars at like 2, 3 in the morning, and then created COVID Poops 19 account around May 2020 and did actually using that to get data, since I saw that that was more recent data than the publications. Publications, even with preprints, take a while to come out, but there was a lot of news stories. So we created the Twitter account to retweet, and it's been like almost 16,000 or so tweets, but I do daily keyword searches with a combination of 6 different combinations: COVID-19, SARS-CoV-2, sewage, and wastewater. And also look and follow some of the major players and researchers in the space to find out where monitoring is expanding. We only report on the dashboard publicly available information through news stories or publications. If we see it on a webinar, we don't just grab it unless it's been like published somewhere else on a website. And we also do traditional literature and internet searches, and there's a Google form at the bottom of the dashboard. We've had over 80 submissions. And then we sort based on 4 categories. If there's dashboards or data available on the website, universities or institutions, and then countries. I just added Iran this week, so we're at 58. And we're close to 3,000 sites. These are sites, even one dashboard might have hundreds of sites, or just like 10 or 11 sites. So we do put the site points on there so people can look at the dashboard and see if wastewater is being monitored in their area. There's a search function on the top right, and you can zoom in and it's active. And we try to update about weekly. We're like a skeleton crew of like a few, like 4 students now, some taking research credits. And all our methods are on the preprint. I do need to update the preprint. It's still in, of course, the review process, but the methods are the same. There's been still, it's keeping up in the space is challenging. There's been a lot of advancements, luckily, and more people are sharing data, and there's still increase in new people starting to monitor. So this is the overall dashboard and Twitter account. We have over 3,000 followers, which is, I think, pretty good for a poop themed account. We're not like some of the major celebrities out there with 10K followers, but it's been, you know, a silver lining of the pandemic to be connected with people that are interested in the wastewater monitoring, and not just researchers, but politicians and just the general public, and they're supportive and they tag us about events or about monitoring that's going on. And we try to answer questions and somewhat dispel like some of the, you know, myths out there about like you getting COVID from your, you know, toilet or wastewater, since it's pretty low risk of being like fecal oral transmission. And so this is the dashboard over time. We launched it in September, and then yeah, over a year, we've doubled like the amount of countries, and the dashboards have really increased, and then we added site points. So it's really kind of, I've seen somewhat of an exponential growth sometimes being monitoring with the keyword searches, and even the baseline. There were people in this space before, a lot of great researchers doing work, and so even before September 2020, or even before the pandemic, there was probably a bit less. And now we've just had a lot of people involved in this space, some that were more stormwater people or involved more in bacteria than viruses. And they really tried to learn and monitor and like help in the pandemic. And so we have a lot of new undergraduates, graduate postdocs, and faculty that are trained in these methods, as well as companies and laboratories moving forward, which is a precursor of how has the pandemic changed the water sector, kind of creating these systems and these networks and these trained personnel. And we've been privileged to, we're still trying to get to that, early warning dashboard with wastewater. It's a bit more difficult than clinical data, that there's so many different methods out there and different concentration methods, whether you're monitoring the influent or the solids, and different quantification and normalization. So getting all the data into like one format and comparing it, it really is difficult. I mean, you really just have to compare trends over time and not just like genome copies per liter, because it really varies depending on the size of your system and such. But so we've become part of the Wastewater SARS Public Health Environmental Response WSphere data center. And this is like a third project of the Global Water Pathogens Project. If you haven't heard of that before, it first started updating the Feacham Sanitation and Disease book. So I was involved as a co-author for how different sanitation technologies remove pathogens. But there's all different chapters of every different pathogen and also indicators. And they also have some knowledge to practice tools and mapping, like a pathogen and a sketcher tool and mapping tools that you can check out. And COVID-19, or the data center, now is the third tool. So you can get there from the GWPP website, or you can go to sphere.waterpathogens.org. And so the mission is to advance environmental surveillance of sewage to inform local and global efforts for monitoring and supporting public health measures to combat disease. And this is a huge effort with PATH, a nonprofit for global health equity, Michigan State University, KWR Water Research Institute, and Ventec Technologies, as well as Merced. And so we do we integrate data from high income country dashboards. If you see the dashboard, there's still a bit of disparity, similar to vaccinations and clinical testing for access to wastewater monitoring. I think it's about 60% high income countries. And so we do integrate that data, but there are field deployments that are supported on the project. So in Pakistan, Nepal, Bangladesh, Malawi, and Ghana. And so we are trying to get that data onto and we have some of it on the data center already. And we wanted to develop some approaches to visualize the data. I emphasize methods in geospatial analysis and geospatial visualizations. So it's great to visualize it in a common format. There's a lot of great dashboards and variety out there, but it's good also to keep it in a different or in a standard format. And then we provide access to the data, and then it's in citable format, and people can download all in one place and hopefully run some comparisons and learn from the data. And lastly, we provide use case studies for public health use case to kind of document how wastewater-based epidemiology for COVID-19 has been used, and to learn from, and also to encourage others to adopt these methods. So this is our data flow, as I said, coming from COVID poops and from the dashboards. There's about half of the dashboards have downloadable data. If you take out BioBot, it's actually just 31 dashboards that have downloadable data, but we harvest that data and we have a data template and data agreements, and we input it in there and there's a lot of back and forth sometimes, and we do reach out and try to have some more regional and geographic diversity. And then we enter it into ArcGIS. We use the dashboards feature and maps, and for the use cases, we use ArcGIS story maps, and we do have a website that the map and the story maps are hosted on. So far, out of those 58 countries, we have 24 countries on the database, 168 sampling sites, over 20,000 samples. So we've been able to put all the data sets into like 12 of them into our format, and then we have 7 more coming in 2022. This is a lot of work by the students and data digging. I know day 2 was about data standardization for this conference, and so yeah, it's still a big hurdle, or making sure like all the methods are in the they don't, when they publish their data, not all the details are there, so sometimes we have to ask for limit of detection, limit of quantification, and things like that. So this is sphere.waterpathogens.org, so you can visit the global map by clicking on the top or on the left-hand side. And so you'll see, similar to COVID poops, but more focused on the dashboards. And then the ones that we've been able to put in our data forms and have all the minimum information we require on, like presence, absence, limit of detection, flow, population, and the methods are in green. And then the ones not quite like standardized or having all the information in our format, maybe don't have the like SARS-CoV-2 like concentration data yet, or don't have flow data, those are in orange. And then we have the grayed out ones, are the ones for future dashboards we would like to include. And we update this about monthly, so it's best to go to the, if you want the real-time data, to go to the actual dashboard. So we're creating more of a repository, but in the future would like it to be more automated. There's a few data sets that we've been able to just automate and update as you go. So yeah, as I said, the website, if you click on a point, you can go to the website of the actual dashboard. Scotland is a great example. They have a lot of great downloadable data and include a lot of detail. And you can see how many sites are monitored, whether it's regional, national, building scale, how many samples and when they began sampling. And then you can go to view sites, and then you'll see the sites that are monitored, and by presence, absence, presence in red, absence in green are not specified, and the bubbles are sized by population monitored. And so you can visualize that. You can toggle a timeline and see over time how the presence and absence has changed. And a lot of these efforts, people are adding sites, so it's interesting to play the timeline and see the number of sites increase. And then when you click on a certain site, you can see what gene is monitored, the population, maximum detected concentration, and average concentration. And then you can go to a trends view, so the sort of population bubbles go away, but then you get the increase, slight increase or decrease with the upwards or downwards arrows and color-coded in red and green, or with no change, and you can still have the trends view at the bottom right. And these trends are calculated based on linear regression of the 5 most recent sites, so what CDC uses. And then you can click on the individual site and see the data over time, like increasing and hopefully it decreasing. And so you can always go back to home on the left-hand side. And if you're not wanting to focus more on the mapping or the geospatial visualization, you can look at the data. We have a place, we hope, if you have data, to contribute data, and there's a data form, and we have our data templates online so you can view, and we can also like take whatever data your format is in and put it in our template. We encourage you to reach out or spread the word. And you can also search the data sets on here by country or by scale. And if you just go to EWOG, you'll see the data set, and you can download the sites. You can download the geospatial layer and the samples layer, and you can see some summary of the data set. And there's also, if you scroll down, there's a link where you can cite the data set, like sort of almost like a DOI number for the data. And you can also view on the map or view on the website as well. So lastly, I'd like to get to the use cases. And so again, you can contribute. There's a contribute form, and then we have our template if you'd like to see. We do focus, there's a lot of great research out there on the methods for wastewater sampling of SARS-CoV-2, but these use cases are focused on how people, or cities or public health departments, are using the data for action. So public health use cases. We have 2 so far, but we have like several in the works that are filling out the forms. So we have a Dutch sewage surveillance case and a Catalan surveillance network. And so these are ArcGIS online story maps. They're like brief descriptions and lots of pictures. So hopefully like visual healing and you can get through quickly without like a huge like paper or report. So the Dutch case is using wastewater to identify areas to increase clinical testing. And the Catalan surveillance network of SARS-CoV-2 and sewage, they had a like 56 wastewater treatment plants monitoring. They have 80% of the population covered and started in July 2020, and they used it as an early warning system and also a complementary indicator for decision making. And it also helped them target clinical testing as well. So we are calling upon like any utilities, laboratory, public health communities, anyone here submitting their data or accessing the global data sets to join the collaboration. So you can, there's a contribute form, and then we also have our water_pathogens Twitter account and COVID poops 19 closely aligned. So moving from this collaboration, I mean, we're kind of creating this data set and this framework, but we are looking into the future. And I know we're continuing to experience COVID-19, but also for hopefully not anytime soon, future pandemics and also other pathogens. And so looking beyond like WB, beyond COVID-19, I mean, the monitoring, people are getting their monitoring extended for about a year or more now, and it's looking with the vaccine disparities and COVID may become endemic, but there's a lot of different pathogens that can be monitored that we could use these systems that have been developed or monitoring networks that have been developed for SARS-CoV-2 to monitor like a larger subset of pathogens and more regularly. There's already, I see a lot of sites monitoring for influenza, and we'll hear more today about Zika and dengue later in the session. And I went to session 7 about antibiotic resistance or antimicrobial resistance, as Michael Gillings had a great presentation about the threat of this and that it might lead to 10 million deaths by 2050. And so really using these systems we've developed, the national, the regional, to monitor for antibiotic resistance. So Virginia Tech, Amy Pruden and Peter Weichsland just had a recent opinion on seizing the moment. Now is the time for integrated global surveillance of antimicrobial resistant and wastewater environments. So how has the pandemic changed the water sector? We've had the EU system or sewer sentinel system, and they're having a next town hall in the coming like month in November, and they've really put a lot of funding, and they're going to have their own dashboard and like collaboration as well. We've seen we've seen other countries like just even within themselves, like Turkey, the UK, France, have a lot of great monitoring and spread out throughout the nation. And then the US has the National Wastewater Surveillance System. I sit on a utilities committee informing this effort, and they're wanting to move forward to continue to expand the system for SARS-CoV-2 and then also other pathogens. And then I just see, it makes me smile that like a lot of politicians have supported these efforts, like really advocating for funding. Don't usually see US like senators, mayors, governors even talking about wastewater, but the pandemic had them focus on that, and also focusing on water in general and making sure everyone has access to hand washing. So it's really improved funding for especially indigenous populations to get them better access to water outside of just wastewater. And Australia has had some interesting debates among their politicians of where the signal or where the poop came from. And even just on popular media, on TikTok, on YouTube, you see some funny things that is just kind of exposing the sector and what we do, which is, I think, important to get some more exposure for wastewater surveillance and water sector. So some key takeaways before I close, just seeing from, as you saw, just the dashboard from a year ago to today, just this rapid expansion of wastewater-based epidemiology, just greater collaboration between water sector, so scientists, microbial ecologists, engineers, public health, utilities, cities and policymakers. There's a lot of different monitoring networks where these people meet weekly and come to the table and discuss the data. So they're also discussing other things and how they can collaborate in the future. So creating those networks and that common vocabulary to communicate to each other. There's still a lot of challenges. There's a lot of people monitoring wastewater and they'd like it to be used more in decision making, but there's a lot of great use cases and that we're hoping to document. And just creation of these national, regional, and global monitoring networks and like a trained workforce moving forward and databases like WSphere. So I'd just like to acknowledge this is a huge effort of different partners listed here and the students and the postdocs, especially for the data center. And thank you so much, and thanks to the sector and all those monitoring that have contributed during this time. And it's still a difficult time, and I know we're all tired and like fatigued by the pandemic, but couldn't do the dashboard and the data center without all those contributing data and monitoring wastewater. Thank you again for this opportunity to give this keynote. Yeah, definitely feel free to reach out on my email or on Twitter at any time if you have any questions or would like to contribute anything. So thank you again.
Thank you very much, Dr. Nocton. It's impressive and encouraging to see how quickly you managed to assemble a team, get funding, coordinate efforts, and launch such world-leading initiative that manages to spread information not only among scientists, but also to the regular citizens. I was wondering, and I would like to start the Q&A session, so at the very beginning, you show how the University of Arizona managed to use the information from wastewater-based epidemiology, not only for surveillance, but also to trace a plan of action and mitigate a possible outbreak. How do you plan to translate all the information of your platform to plans of actions through all the dashboards that are part of the initiative? And which role do you think scientists can have there, and especially you and your team?
Yeah, no, thank you for your comments and the question. I think through the data center, we're just trying to document in the public health use cases through those story maps of different examples. And I mean, scientists can do this not just through peer-reviewed publications, but on interviews for news articles and just creating more accessible videos and methodology for how they did it. Like University of Arizona had been doing WEBE prior to COVID, and so they had a very good working knowledge of their sewer system, so they're better able to estimate the number of students that could have been infected. So understanding that, since it's still a little difficult and sewage and is messy, as we know in the data. So I think just better documenting and continuing to promote and also through social media is important about these efforts and how they can be useful.
Yeah, there's also an interesting point by looking at poo, and when you have this name COVID poops 19, I don't know how it is perceived by the local public. It remembers me once a presentation from a scientist in France at École Centrale de Paris, Emmanuel Adler, and he mentioned historically how disease were looked at, and if you look at in books, all books, you see medical doctors just take the cup where there is poo, and they look at your poo and they see what disease you have. So they could, just by looking at the poo, detect, okay, you have this or this disease. So now we have much more advanced techniques to do that. They are sequencing on other qPCR measurements, etc. But it bridges us back also to Arden Lockett and other types of medical approaches in the past, where there was not all this sequencing, but poo is an important component of urban challenges, actually. Yeah, and I remember a talk as well of Dulai Kone from the Bill and Melinda Gates Foundation, mentioning that we should try to find sanitation systems that avoid transporting pathogens across cities. It can be viruses, but it can be also bacteria. And how do you think that this platform could help in the sanitation decision as well, for sanitation actions?
Right, great question. Well, first to poop, I mean, I've tried to use like the play on words or make it more attractive to the public. I have gotten some criticism of like, oh, we should use feces or the technical term, but I think it's playful and, you know, something everybody connects to or everybody poops. So just try to make it less of a, you know, stigma against wastewater or poop. Like we really should be paying attention to it. And it's been this important public health tracking tool, and also, you know, just for yourself to pay attention to for your own personal health. So we need to stop being so embarrassed about it. I think for using this tool moving forward for just regular pathogens and removal. We had some of that with like the knowledge to practice tools and like wastewater sketcher tool to kind of like estimate pathogens and like where they're coming from and like the removal of different types of treatment. So we had some of those built before, but it would be good to like expand that. I think it was more focused like regionally in Uganda and other countries, but others can use the data as well. So just, I think there's a lot of power in these tools and like knowledge about how much pathogens are removed and how we can improve in monitoring over time. As far as like, we shouldn't be transporting at large distances. That's always a trade-off. Like decentralized technologies or treatments aren't always the best maintained. So centralization might be best, but making sure it's contained and not spreading to the public, and is also redundant so that if there's a large rainstorm or there's a blockage, it's not creating, or a natural disaster, unfortunately with climate change, not creating these like vectors or outbreaks.
There is a question in the chat from Claudia Echeverre. Is there a big conclusion? You want to go? Yeah, go for it.
Go ahead. Sorry. Sorry about that. Is there big conclusions about monitoring SARS-CoV-2 in wastewater? I mean, we're still wrapping our heads around that. I think with the use cases, and then, you know, a lot of this has been promoting that sort of early warning system, but we're seeing like that can vary over time, and we're still like learning the science and what it can be used. But I think big conclusions are just that it can be a powerful tool, especially for a virus that has a lot of asymptomatic spread, and that we can use this in combination with other testing and mitigation measures. It's not like, I think a lot of articles are trying to make it like the silver bullet, or even when I interviewed, they're just like, oh, we should just use this in place of everything. But you really, if you looked at the Swiss cheese model for preventing COVID, it's like another tool in the toolbox to be able to monitor the spread in the community. And if you do sewer shed monitoring or neighborhood monitoring, being able to look at where there may be outbreaks to target clinical testing or vaccination, but do that, of course, in a mindful way that you communicate to the public and not scare them or feel that they're being spied on.
There's a question in the chat, and I think in the room as well. So in the chat from Tom Curtis, can we estimate prevalence from wastewater?
By prevalence, Tom, do you mean like cases, like case numbers? You can answer, but we are able to give a round estimate. It's not
cases per 100,000.
Can you repeat?
Can we the number of people per unit, number of cases per unit of population?
Yeah, I mean, the community, there were initially some companies like promising that or trying to estimate, but as you saw with the University of Arizona, they are able to estimate the building and even their level if you have a good like knowledge of your sewer system and the flow, and also how much is shedding, like the shedding rates per population. You can get to estimates, but long answer, but not there's like a bit of uncertainty. So we've seen more, it's good to monitor trends over time. So if you see an increase start to see an increase, then you need to maybe have more like mask mandates or target more testing or more vaccination. So it's really good temporally over time. We're still working out the details on like number of per 100,000, though we can get some estimates if you have greater information, and especially more information about the virus. It's been challenging with the variants because some of the variants may have higher shedding rates, so it's just always a moving target of like how to estimate.
David, you had a question on site. Eric, we need the sound up on the stand microphone.
No? Okay. Hello, Colleen. Thank you very much for your presentation. It was great. I love the initiative and the work, and I also love the Twitter account name, so keep it like this, please. I was just wondering, like in this Sphere initiative that you're having with different institutions and projects and people, is there any consensus on how you want, as I guess, to receive the data in terms of quantities, numbers, and is there any way that you have agreed on the Sphere community on how do we need to isolate and report the data from wastewater and from which samples?
Right, great question. I mean, we have developed a data template where we are doing genome copies per liter. We've seen it in like genome copies per milliliter. Of course, that's an easy conversion, but some are doing per pepper mild monovirus as the fecal indicator. So we have expanded another worksheet for calculations, but we are trying to put it in genome copies per liter. And we're trying not to be too, like have too many fields that it's really difficult to report all those values. There's still some, the community is still trying to agree on like what's the minimal reported data. I mean, CDC's news format is really detailed, and then the Europeans have their own data template, and then there's the Norman Score database, and Canada is having an ODM, like open data model, which is I think better using more like programming or GitHub and less moving away from the Excel spreadsheets, but that's a lot what people are comfortable with. But to answer your question, yes, we've had a data form like of what we would like in the data, but we acknowledge that there's like other variations, and we're always trying to learn, and we're like working with those other data centers or formats and how we can translate them into our format. But it's still like a moving target. It'd be great if like it was set up from the beginning and everybody was following a similar format, but the methods are changing so much all the time that that's kind of difficult to do. So we're trying, but don't want to be too restrictive of like, this is the right way.
Okay,
yeah.
Thank you very much.
Thank you very much. We will continue the discussion later in the panel because there are still some questions, but we have to move on. Our next speaker is Diana Patricia Verent from the Instituto de Investigaciones en Ingeniería Genética y Biología Molecular in Argentina. And the title of the talk is A Simple and Rapid Protocol for SARS-CoV-2 Concentration from Sewage. We will launch the video right now.
My name is Diana Verent, and the title of my work is A Simple and Rapid Protocol for SARS-CoV-2 Concentration from Sewage. SARS-CoV-2 detection in wastewater samples can provide information about the prevalence within a community, the emergence of new outbreaks, and the efficacy of the vaccination campaign. As it can be seen on the map, many countries are already monitoring SARS-CoV-2 in wastewater. Viral detection can be performed in the same way as for clinical samples, although wastewater samples have to be concentrated first. The most commonly used methods for virus concentration are laborious and require specific equipment, such as filter cartridges, electronegative membranes, high-speed refrigerated centrifuge, or even ultracentrifuge. This equipment is not always available in every laboratory. In low and middle-income countries, a central sewage network is not always present. Instead, several smaller treatment plants are found. Our aim was to set up a simple protocol to concentrate SARS-CoV-2 from sewage, which can be performed in laboratories with minimal equipment. The final concentration method that we have developed is the following. Grab samples are first heat inactivated at 60 Celsius degrees for 90 minutes for operator safety. 40 milliliters are then transferred to a conical centrifuge tube, and 200 microliters of a diluted polyaluminum chloride solution are added. After 15 minutes of incubation at room temperature, samples are centrifuged at 1700 G for 20 minutes. Finally, after discarding the supernatant, the pellet is suspended in 200 microliters of phosphate buffer saline. RNA extraction and SARS-CoV-2 detection are then performed in the same way as for clinical samples. To estimate the sensitivity of SARS-CoV-2 detection with our proposed concentration method, we artificially seeded inactivated SARS-CoV-2 virus to a negative sewage sample. Serial dilutions of this sample were performed, and SARS-CoV-2 was detected by RT-qPCR after sample concentration and RNA purification. We were able to detect approximately 100 viral copies per milliliter with our concentration method. To test the performance of our proposed concentration method, grab samples were taken weekly at 4 different pumping stations located in the Buenos Aires metropolitan area. Biochemical oxygen demand of the samples varied between 250 and 630, and the pH was 6.4 for SF and around 7.5 for the rest of the samples. Despite the different characteristics of the samples, the concentration method could be successfully applied to all of them. To assess the efficiency of the concentration method, samples were taken on duplicates and concentrated versus non-concentrated samples were compared. An average shift of 4.4 units in CQ values for N gene detection and 4.6 for OF1 gene detection was observed, indicating a 25-fold increase in detection sensitivity in concentrated samples. After concentrated, samples could be stored refrigerated for at least 7 days without affecting the RT-qPCR results. We also compared our concentration method with the one based on polyethylene glycol precipitation. Out of 60 total samples, 45 were positive by both methods, and 9 were negative. Only 6 differed between both methods. A kappa index of around 0.7 indicated substantial agreement between these precipitation protocols. In summary, our proposed method is very simple and fast. It only takes 45 minutes. No expensive equipment is required. Concentrated samples can also be stored refrigerated for at least 1 week without a loss of sensitivity, and therefore be sent to a diagnosis center for SARS-CoV-2 detection. In conclusion, this low-cost protocol could be useful to aid in the monitoring of community circulation of SARS-CoV-2, especially in low- and middle-income countries, which do not have massive access to support from specialized labs for sewage surveillance. Thanks to everyone who contributed to this work and to the Ministry of Science, Technology and Innovation for the funding. And thank you for your attention.
Are you online, Diana? Thank you for your presentation. If Diana or Leonardo are online for some questions. Sergio, can you check if Diana is online on the list? So, Diana Verand. So,
Yes.
Yeah. The question that I would have, it's how do you plan the deployment of your method now? Now you have a method that seems to be affordable, low cost, and that can be implemented in LMICs. How can it be deployed? What should be the strategy to this end?
Yeah, so that's a very good question, and it's kind of difficult. But yeah, the idea is to propose the method for different cities or small communities so that they can adopt it. We also try to do it. So some cities already started doing it, but it's not so easy to do this. But we are working on it. So the idea is that almost a lot of cities can use the method and then contribute with the monitoring of SARS-CoV-2 and wastewater.
Thank you.
There's a question in the chat from Colleen. What is the sensitivity of the method? And also, is the protocol available on protocols.io?
Yeah, so we were able to detect around 100 copies per milliliter using this method. This protocol, we already published it, but I don't know if it's not on protocols.io, but it's published in a journal.
Okay, thank you very much. Let's move now to the next speaker, who is Xuan Lin from University of British Columbia, Canada. The title of the talk is Assessing Multiplex Tiling PCR Sequencing Approaches for Detecting Genomic Variants of SARS-CoV-2 in Municipal Wastewater. Are you online, Xuan Lin?
Yes, I'm trying to share my screen. Where does that?
Do you get an error message or not? So there is, if you
yeah.
So what do you get on your screen?
I'm getting screen share failed because I only got 2 screens available to share, and none of them is my slides. So, do you have any suggestions for how to sharing?
Sometimes when you have 2 screens, sometimes it gives an error. So is there a possibility that you unplug the laptop maybe from the other screen so you only have one monitor with you? Maybe like that it won't give the error.
Let me see. Windows. That's can you wait a sec? I think it's some settings there.
Sometimes there are some problems and you have to go out of the platform and rejoin the platform. Or Zeb, do you have any idea what can be the solution?
Could you wait a sec? Because it asked me to quit and reopen the Google Chrome right now. So
okay. Maybe we move to the next presentation and we come back to you after. Okay?
Okay. Our next speaker is Colleen Jacob from Veolia Environment Research and Innovation. And the title of the talk is Reclaim Water Quality Regarding SARS-CoV-2 and Other Microbiological Parameters in the Framework of the Development of a Smart Decision Support Tool for Sparkling Fertigation of Arable Crops, Smart Ferti Reuse. Video coming.
Hello, I'm Pauline Jacob from Veolia France. and I'm going to talk about weak water quality regarding several microorganisms in the framework of the development of a smart decision tool used for fertigation of crops named Smart Ferti Reuse. In a context of increasing hydric stress, the objective of this project is to promote reuse and anticipate future regulations. Smart Ferti Reuse is a collaborative research project that aims at developing a new decision support tool to provide safe and optimal management of water and fertilizers for agricultural irrigation, taking into account crop specificities. The project was conducted on a test site located in the south of France. The objective was to monitor the water quality produced from 2 tertiary treatment pilots combining different treatments, downstream a municipal wastewater treatment plant, the first one for A quality and the second one for B quality. Analytical campaigns were performed on reclaimed water and on the groundwater that is currently used for irrigation in order to compare their microbiological quality. The water quality was also benchmarked against quality requirements in French and European regulation for water reuse to assess treatment performance and obtain data for risk assessment regarding microbiological contaminants. French and European regulations impose requirements in terms of microorganisms log reduction, with higher requirements for quality A than for quality B, shown in this table. This table shows microorganisms reduction obtained during 2021 campaign on Channel A regarding French regulation. The performance of the Channel A meets the requirements of the French regulation. On the same way, the performance of the Channel B also meets the French regulation requirements. Regarding European regulation, the performance of Channel A meets the requirements except for spores, which not meet the reduction threshold required, but this does not block irrigation because it meets quality B level. Furthermore, on this stage, we are only in an anticipation phase of the European regulation. This table shows French and European requirements in terms of concentration objectives for E. coli for both regulations, with higher requirements for quality A than for quality B, and regarding Legionella and helminth eggs in European regulation. 25 samples were collected from March to September. For A pilots, 19 samples concentration were below the detection limit. 6 samples were positive, 5 below the threshold and 1 above, most likely due to a sampling problem. For B pilot, 23 samples were below the detection limit, 2 samples were positive, below the threshold concentration. So reclaimed water quality complied with the French and European requirements. This slide shows results obtained for parasite monitoring at different stages of the treatment. Point detections can be seen for total giardia at the pilot outlet with the maximum quantity below the infectious dose. The most important being that not a single parasite was detected either at any pilot or pipe outlet. There were no Legionella found by culture. The risk of contamination is therefore limited in case of inhalation. No helminth egg was detected at the pilot outlet. There is only 1 positive point during all campaigns at the wastewater treatment plant inlet And at the same date, the pilot outlet was negative, but with an associated positive point at the pipe outlet, but with very low concentrations, so with a limited risk in case of ingestion. This positive point could be due to a contamination during irrigation. There is occasional presence of Salmonella at wastewater treatment plant inlet and outlet, but never detected at pilot outlet, and absence of Campylobacter in all samples. So the risk is therefore limited in case of ingestion. In 2020 and 2021, in the context of sanitary crisis, monitoring of SARS-CoV-2 by PCR was carried out on reclaimed water, independently for any regulation. Indeed, little is known about the effectiveness of wastewater treatment on the virus, but the potential exposure of neighbors of the plot to aerosol induced by irrigation is real. This graph shows the monitoring of the virus at different points in the treatment chain, with concentration threshold defined according to the quantification limit of the system. The detection at wastewater treatment plant inlet was comprised between levels 1, 10 quantification limit, and level 2, 100 quantification limit. Same levels were observed in 2020. There was no detection in wastewater treatment plant outlet or pilot outlet in 2021. Some rare positives were detected in 2020, but at the detection limit. It can be observed that there was a reduction of SARS-CoV-2 load in wastewater due to wastewater treatments. So virus reduction and therefore the risk related to its aerosolization are reassuring. This slide shows an example of groundwater monitoring used in conventional irrigation. The concentration measured in the groundwater in 2021 are close to those previously measured. Microorganisms are naturally present in the groundwater in varying concentrations depending on the season and year, but in low concentrations. In 2021, no parasites were detected. For the first time in 2021, selected agricultural fields growing cereal were irrigated with reclaimed water to assess potential impacts on the crops compared with conventional groundwater irrigation methods. Results are under analysis. Reclaimed water quality complied with French and European regulation requirements. In terms of microorganisms removal and threshold concentrations, and there is a limited risk in terms of ingestion or inhalation for other microorganisms. There is a reduction of SARS-CoV-2 load in wastewater due to treatments, then the risk related to its aerosolization are reassuring. I would like to thank all of the project partners and thank you for your attention. Feel free to ask questions.
Thank you, Pauline, and also Adele for the talk and the very interesting initiative. So you clearly showed how the project compiles with the sanitary requirements. Can you anticipate any problem for crop growth? And also, what are the next steps for you to be able to use this water?
Okay. Hello, thank you. I'm answering question because Pauline couldn't join. So I think the point with ferti irrigation is more ferti irrigation than the risk using this water. The one other aim of the project is to evaluate if reclaimed water can enhance arable crops growth. So I think, as it was shown in the presentation, in terms of risk, it's really it's more than okay. And yes, the next steps are to evaluate the interest of using such water for fertigation of crops. I hope it answers the question.
I have a question about the survival of the virus. So is it an active form or is it inactive? For irrigation, what happens if you have inactive virus particles? Is it a problem? Is it not a problem?
We cannot say that. We cannot answer this question from PCR data. So I don't know, but the literature on the virus infectivity is go more in the sense that there is no, or at least low concentration of infective virus at the inlet. So I cannot definitely answer, but my feeling is that the risk is limited.
There's a question in the chat from Colleen. Do you think there may be a risk for other pathogens than SARS-CoV-2?
Good question. Yes, definitely. Exactly. I think COVID has highlighted the risk of yes, using wastewater, but some other pathogens were already in wastewater before the COVID crisis, and we would like to search for other pathogens, but today I cannot answer really. Again, in the perspective of using reclaimed water with the membranes and everything, the risk seems limited.
Okay, thank you. Maybe one last question from me. Usually the concentration of everything in groundwater is very stable because the residence time is very high. But you showed it's not really the case here. What is the residence time of the groundwater you're using?
The groundwater that the agriculture are using, you mean?
Yeah, so usually when you use groundwater, it stays there for years actually. So the concentration that you get when you abstract it is always the same. But you showed this is not the case. Why so?
Yes.
Is it not there for long enough?
Okay, maybe it was not that clear, but groundwater is not the main topic of the. We want to use reclaimed water, and the comparison was done with groundwater to see the microbial containing yes the microbial inside the groundwater. I don't know. I can ask Pauline if she knows. She is able to answer your question, because to be honest, I was more involved in the COVID part than on the global project. But I will ask her and keep you informed.
Okay, thank you very much. Let's move now to the next speaker. No, let's go back to Xuan Lin. Are you still here?
Yes, sorry for the technical issues, and thank you for all your patience with that. I'm wondering, is that looks good right now?
Yes,
yes, we can see your screen.
Okay, and I will try to see if I can move my screens. Yes, it looks good. Okay, can I start my presentation now, or?
Sure, floor is yours.
Okay, thank you, everyone. And I'm really glad to presenting my work. I'm Shelling from the University of British Columbia, and I'm really glad to present our work on assessing multiplex telling PCR sequencing approaches for detecting genomic variants of SARS-CoV-2 in municipal wastewater. This work is in a collaboration with BC Center for Disease Control and Metro Vancouver. So thanks, Dr. Nocton, for a detailed introduction to the wastewater-based epidemiology. This wastewater-based epidemiology was first developed as a potential tool to evaluate the use of illicit drugs and misuse therapeutical drugs in 2001. It is an inexpensive and near real-time analysis tool and has been used for monitoring the community transmission of SARS-CoV-2 since the beginning of the pandemic. About half of the patients excrete detectable levels of SARS-CoV-2 virus inside their feces, which will end up in wastewater treatment plant through sewage pipelines. By detecting the viral signals inside wastewater, we could therefore estimate the community infectious levels. Since the December of 2020, several emerging lineages of SARS-CoV-2, so-called variants of concerns, has been associated with increased level of transmission, decreased severity, disease severity, and immune escape. Previous study has shown that RNAs from wastewater can be sequenced to look for specific coronavirus mutations or lineages. Therefore, genomic surveillance there plays a critical role in monitoring the community transmission of SARS-CoV-2 variants. An important characteristic of SARS-CoV-2 virus in wastewater is its relatively low viral titers. Therefore, an enrichment step is typically needed parallel to sequencing to improve its sensitivity. One of the key enrichment methods is multiplexing PCR-based target amplification, as shown in the figure here. Multiplex telling PCR is a combination of PCR primers for amplifying overlapping amplicons, which will span the whole genome of SARS-CoV-2 virus. An important consideration for applying multiplex telling PCR method is the average amplicon length, as this may impact the assay sensitivity, especially in the cases of wastewater-based epidemiology, as SARS-CoV-2 particles and free RNAs can undergo various levels of degradation and may differ based on the sample matrices that say influent, wastewater, or primary sludge. We therefore hypothesize that there might be an optimum tiled PCR primary schemes and an optimum wastewater sample matrix, which will enable adequate genome coverage of SARS-CoV-2 for identification of genomic variants of concern. The objective of this research is firstly assessing the performance of 3 different multiplex telling PCR primary schemes, which are 150 base pair amplicon schemes from Swift Biosciences, 400 base pair primary schemes developed by Arctic, and 1200 base pair amplicon schemes developed by Fred et al. Secondly, assess the performance of 2 wastewater sample types, which is influent wastewater and primary sludge, both for identifying the genomic variants of concern in wastewater. Our approach to sequence SARS-CoV-2 in wastewater was first extract DNA from wastewater samples, then quantify them with RT-qPCR. This part was done by our partner at BCCDC. The SARS-CoV-2 RNA was reverse transcribed and enriched with 3 different multiplex PCR primary schemes. The 150 base pair primer amplicons were sequenced on Illumina sequencer, while the 400 base pair and 1200 base pair amplicons were sequenced on nanoporamine ion sequencer. The performance of each primary scheme was assessed, and the best performing assay was used for monitoring the variants of concerns in Metro Vancouver wastewater. We sequenced 96 wastewater samples collected between February and April 2021. The figure here shows the breadth of coverage, genome coverage for influent and primary sludge samples sequenced with different primer schemes. The circled green dot here represents the sludge samples. All such samples failed to produce libraries with higher than 32% of breadth of genome coverage across all different primer schemes and CT values. Conversely, influent wastewater samples produced libraries that had significantly higher breadth of coverage across all primer schemes. Since no inhibition was detected with the internal controls for sludge samples, it is more likely that RNA inside the sludge samples are more degraded than in the influent wastewater, and also a higher off-target amplification in sludge extracts. In the other way, if the level of RNA degradation within a wastewater sample will impact the resulting SARS-CoV-2 genome coverage, we would expect less of a drop-off in coverage at high CT values for shorter amplicons. Indeed, that drop-off in coverage was observed for samples enriched with 150 base pair primer schemes. As shown in panels D to F, the median genome coverage with the 150 base pair primer schemes span one order of magnitude, with CTs ranging from 31 to 37, while those for 400 base pairs and 1200 base pair amplicons spanning over three orders of magnitude for samples having CT ranging from 31 to 37. Thus, shorter amplicons primer schemes might be more robust to RNA degradation with higher CT values. However, there was a trade-off between the amplicon length and the genome coverage, as the magnitude of medium genome coverage as well as the breadth of coverage obtained for samples enriched with 150 base pair primers were significantly lower than those obtained with both 400 base pairs and 1200 base pair amplicons, which is likely due to the complexity of the 150 base pair primer pool. Therefore, sequencing with the 400 base pair primer schemes and influent wastewater appear to be the most suitable approach. In order to estimate the frequencies of VOCs in wastewater, we generated lineage-specific single nucleotide variant tables, in short, SNPs tables, for each VOCs we studied. These tables were generated by mapping sequence from P1, B117, and B1351 lineages available from GIS8 against the Wuhan reference genome. Then select the SNPs that have above 90% frequency in each of the lineage, but having less than 10% frequency in all other lineages. In total, we have 26 SNPs selected for B117, 11 SNPs for B1351, and 17 SNPs for P1 lineage. With the lineage-specific SNP table, we were able to estimate VOC-associated mutations from wastewater. This figure shows the P1 signal in wastewater samples during the study period. Each colored dot represents the frequency of a single nucleotide variant, and the black dot represents the mean frequency of the P1 associated mutations. This time series clearly shows that the emergence of P1 in March 2021, and then the frequency increased to a maximum mean frequency of 40% across all wastewater treatment plants. The frequencies of wastewater P1 associated SNPs align well with the results of clinical screening and sequencing of patient samples, as shown in the left figure. The P1 lineage started to be detected in mid-February and reaching about 40% at the end of our study period. The figure on the right shows the correlation between wastewater P1 load and the clinical P1 cases. The log transformed flow normalized daily load P1 loads across all wastewater treatment plants were also strongly correlated with the case counts from the corresponding epi weeks. Therefore, the frequency of VOC-associated SNPs within wastewater within influent wastewater measured with multiplex telling PCR is a suitable approach for monitoring community transmission of genomic variants in a sewer shed. Here are a few key takeaways from our study. Firstly, the RNA extract from influent wastewater provided higher genome coverage than from primary sludge. Secondly, shorter amplicons were more resilient to sample RNA degradation, but were hindered by greater primer probe complexity. Thirdly, the optimum primer schemes, which is 400 base pair primer schemes, and the optimum sample matrix, which is influent wastewater, could detect the emergence of mutations associated with VOCs, and it is correlating strongly with our clinical case counts. To be noted that we were able to have a turnaround time as short as 3 days with our approach. Therefore, the rapid turnaround and low cost of this approach makes it appealing for routine genomic surveillance of community transmission. Lastly, I would like to thank our partner of BC Center for Disease Control and Metro Vancouver, and also thank NSERC for funding this research. And thank you all for your attention.
Thank you, Xuan. There is a question from Changji Wang. Thank you for your great presentation. Is P1 the only VOC found in your samples? Are these as in
yes.
Second part of the question. Are these SNVs from the whole genome or just the spike protein?
So to answer your first part of the question, P1 is just an example. We monitored 3 VOCs which appeared during our study period, which are P1, B117, and B1351. During February and April 2021, there is very low detection of B1351 in British Columbia, but like the cases were dominated by P1 and B117. However, due to the time limit of this presentation, we're only showing P1 results as an example, but the other, the B117 results are also quite similar to what's presented as P1 and showing clear trends and strong correlation with the clinical cases. To answer your second part of the question, we are sequencing the whole genome, not just the spikes protein.
I have some question related to this. So on your slide 5, you speak about genome coverage, but it seems that you only target one gene, so the gene N. So I didn't catch in your presentation, you speak about genomic surveillance, you speak about genome coverage, but apparently you want to make a multiplex PCR. So it would mean that you cover many regions of the genome. Do you want to cover the whole genome? So you fragment the genome in small pieces, so you amplify small pieces and you want to reconstruct the genome, or you are targeted single genes and you do amplicon sequencing in the end on some genes? That I didn't understand.
So let me come back to this slides if I'm not really explaining it clearly. So the example of these short bands are just one of the fragments of the genome, which showing as an example. But in fact, this short primer, the primers were amplifying those short fragments across the whole genome. So for the 150 base pair primers, they are amplifying over 300. They are having over 300 primer pairs, which amplifying over 300 different short amplicons. And for this Arctic 400 base pairs, they are amplifying over 100 amplicons across the whole genomes. And the same for the 1200 base pair primer schemes.
Thanks. And a follow-up about the RNA. Do you use purified RNA or raw RNA? Because it seems that there is an important effect on whether you include a purification step or not on the sensitivity of your method. So David tested that in the past.
So the answer is no, we're not really using any purification in between the RNA extraction and sequencing. So our workflow is going with extract the RNA from the sample, and we use this RNA extract to do our downstream reverse transcription and PCR amplification. However, in the RNA extraction, we were using a MAC-based extraction method, which gives us quite clean of the RNA extract, and we detect no inhibition in both the influence samples and primary select samples. However, to be noted that there are some RNAs from other genomes and other species, because we are extracting the total RNA, not just the SARS-CoV-2 RNA from the wastewater. Therefore, there are some off-target amplification with this method as well.
Thank you.
There are still a couple of questions, but we will move on to the next presentation and talk about them during the discussion panel.
Thank you. Yeah, thank you for the questions, and yeah, thank you.
Thank you for the talk. Our next speaker is Francis Chandra from Singapore MIT Alliance for Research and Technology. And the title of the talk is Persistence of Yellow Fever, Zika, and Dengue virus in untreated wastewater.
Are you online, Franciscus? Or it's...
Presentation slides.
Yeah, you can see everything.
All right, great. Okay, good evening, everyone. Thanks for tuning in, and thank you for the organizers for providing the time. It's really a great honor to be speaking in this conference. My name is Francis, and today I'll be sharing briefly about the research on persistence of arboviruses in untreated wastewater. So a bit of a background. This research is part of Singapore's focus into wastewater-based surveillance. In particular, the arbovirus work is a collaboration between Smart MIT and CELSI NTU. But we really are a multi-institutional group with collaborators, ranging from Duke NUS, NUS, and NTU. So some of us might be wondering, what are arboviruses? So arbovirus is a shorthand term of arthropod-borne viruses. And they're called that because they're mainly spread through arthropod vectors, such as mosquitoes, ticks, and sandflies. They are a polyphyletic group consisting mainly from the genus Flavivirus. And most of them are single-stranded RNA envelope virus. I would like to turn the attention now to this global map of arboviral spread. And so you can see here the arbovirus is actually endemic in multiple countries, and then they incur high burden of disease. For example, dengue fever here has almost 400 million infections per year with 9 billion healthcare costs. Yellow fever, around 200,000. West Nile virus, which may be more interesting to those in the Northern Hemisphere, has one of the most broad endemic region for arbovirus. And of course, there's also the Zika virus with its infamous outbreak in Brazil in 2015 and 16. Another aspect of this arboviral cases, so to say, is their asymptomaticity. So large proportion of arbovirus is actually asymptomatic. For example, dengue virus, some have estimated as high as 75%. For yellow fever, 55%. And for West Nile virus and Zika, as high as 80% of them are asymptomatic. And we might be thinking that, isn't it great that most people that get the virus are not getting sick? Well, not really. The problem with these asymptomatic people is they can still transmit the virus to the mosquitoes, the main vector. And in turn, they can still infect other people. And so some modeling has been done, and some people have claimed that these asymptomatic cases play an important role in arboviral persistence in a given geographical region. So here we have like a family of disease that is like widespread, have high social economic burden. It's hard to survey because the majority of them are asymptomatic. And also with the rise of global temperature, climate change, we expect an increase of the geographical reach of these vectors to more subtropical regions, making this like a very pressing public health issue. And so how do we tackle this? One interesting thing that we found about like arboviral infection is that they are shed in the urine. So people have found like Zika virus in urine. People have found dengue virus in urine. And recently, we tried to compile these shedding rates in a review paper. The data is quite scarce, but when we compare them, say, with the more mainstream viruses with successful wastewater-based surveillance programs like SARS-CoV-2, poliovirus, norovirus, we can see here that these arboviral shedding rates actually overlap. And this gives support to the possibility of arboviral wastewater-based surveillance. Now, for arboviral surveillance, currently there are kind of like three pillars relying on their complex natural cycles between their natural reservoirs, their insect vectors, and their symptomatic human hosts. We want to propose wastewater-based surveillance as this fourth pillar, as a complementary surveillance system that can hopefully, capture the symptomatic and also the asymptomatic cases. And this development of wastewater-based surveillance can hopefully lead to a more rapid policy response, and thus we can prevent, another 2016 Zika outbreak early on. And so with this backdrop, we conducted our study on the persistence of these arboviruses in wastewater. We aim to determine if these arboviral signals persist long enough for sampling, processing, and analyzing. And so this is the overall experimental workflow that we did. It's pretty simple. We took a panel of viruses, which include the arbovirus, yellow fever, Zika, and dengue. And we also have a murine hepatitis virus as a point of comparison with other studies. And then we incubate them in 3 different temperatures, 6 degrees, 25 degrees, and 37 degrees. These temperatures were taken because the 25 degrees and 37 degrees is a proxy for tropical, subtropical region. And the 6 degrees is kind of like the temperature where we store wastewater sample. We tested them across 21 days and through RT-qPCR to determine the genome copies. So this is the data that we got out of it. And just to summarize this big table over here, what we found amongst the arboviruses, Zika and yellow fever have roughly the same decay rate with a T90, or the time needed for 1 log of decay, around 2.5 days to 4 days, while dengue T90 is around 3 to 4.5 days. And all of these arboviruses are stable at 6 degrees Celsius. As part of our study, we also looked into the effects of heat inactivation on arboviral wastewater samples. So interestingly, for samples that are heat inactivated and then directly processed, we see that there's a slight increase for the signals for some of the viruses. Most notably here is the yellow fever and the murine hepatitis virus. We hypothesized that the heat inactivation here helped with either the viral lysis or detachment from suspended solids. And for all the other arboviruses like the Zika and dengue, there's no significant difference, meaning that heat inactivation doesn't affect the signals that we get out of the samples. However, for samples that are heat inactivated and then incubated at 6 degrees, we see that heat inactivation increases the decay rate. And this is, to be expected because heat inactivation would disrupt the viral capsid and all other things. However, this data is not statistically significant because the little decay that we observe at 6 degrees. And the experimental noise here kind of outweigh the decay signal. So what is the implication of our study over here? First of all, a majority of arboviral RNA signals seems to persist long enough for sampling. And we can take like 2 timings as our benchmark. The first one is like average sewage residence time. And we'll take here as Singapore as a benchmark for a few hours of residence time. And the signals persist long after that. Secondly, the average wastewater sampling time. Most auto-samplers are set to 24-hour composite. So during this sampling time, we expect around 30 to 40% signal decrease. And to help with the signal decrease, to prevent further signal decrease, refrigeration can preserve the signal. And it might be worthwhile to implement in future workflows for arboviral wastewater-based surveillance. And finally, heat inactivation of wastewater samples prior to processing does not seem to decrease the signal generated. But caution is advised if you want to heat inactivate samples to be stored long-term. So for our lab currently, we are working on to run pilot tests in Singapore, mainly on dengue, because it's quite prevalent here. But in the overall arbovirus wastewater-based surveillance, this is only one part of the puzzle. There are many other factors that needs to be researched further. Decay rate is only one part, whereas we also need to think about your assay sensitivity, since most of these molecular assays are made for clinical samples and not wastewater. We also need to think about the cost-benefit ratio of actually doing these arboviral surveillance using wastewater. And we can also think about the recovery rates from wastewater. So with this, I end my presentation. Huge shout out to the supervisors of this project, Janelle Thompson from NTU and Eric Alm from MIT. Also, shout out to the team at Smart AMR and Celsi NTU, who also stayed up late and to be in this presentation with me. Thank you for the funding agencies, the Singapore National Research Foundation, Ministry of Education, and the Create Campus for their support. Thanks for listening. And any questions?
Thanks for your motivation as well. It's fantastic to see a very lively presentation from you. Thank you.
Thank you.
It's midnight, I think, in Singapore, if I understood.
Oh, yes.
Thank you very much, Francis. I can't agree more with David's statement. There's a couple of questions from Colleen. The first one is, what about sewage travel time impact on decay rate? Some larger systems, it can take a full day to get to a treatment plant.
Oh, that's a great question, actually. Our study currently only uses quite an upstream wastewater because based on our calculations on the prevalence of, say, these arboviruses and also the shedding rates, we kind of determined that they are more suitable for neighborhood-based surveillance. And so we haven't really checked into downstream wastewater, the wastewater treatment plant wastewater matrix. But I can foresee that as it gets more diluted, maybe there is less effect on the decay as it gets more downstream. However, yeah, the question is still up in the air.
Thank you. And another question. Could any of these arboviruses be used as an indicator for the spread of malaria, which isn't a virus?
Wow, okay, another great question. Can it be used as an indicator for malaria? I am unsure of the if we can actually use this as an indicator of spread of malaria, because the vectors for, say, arbovirus and malaria is quite different. But I mean, I could imagine that this could be as a proxy, or there is some kind of correlation between arboviruses and malaria. and definitely an interesting research question to look at in the future.
Thank you. Perhaps it would be interesting to make a wish list of which of the characteristics that a virus or a bacteria needs to have to be able to be followed by wastewater-based epidemiology. We can discuss it later. Let's move to the last speaker, who is, let's see if I do this, Arnaud Deschênes from DTU. The title of the talk is Distribution and Abundance of Plasmid-Specific Bacteriophages in Wastewater Systems.
Arnaud Deschênes.
Thank you.
Yes. I will share my screen in a second. I think I need to close a few windows first.
We cannot hear you hyper loud.
Okay, yes.
I'll
speak up.
Microphone. Yep.
Better now?
Bit. Can you share your screen, or you have a error
Yes,
message?
that's what I'm trying to do at the moment. Somehow I cannot really click on the allow box.
So you
Okay,
do you see the
no,
box?
I think I'll find a way.
Something is.
How does that look?
Perfect. We can hear you. We can see the screen.
Brilliant. All right. So yeah, my name is Arnaud Deschênes. Easier to pronounce for the French people, obviously. And I'm going to talk. Well, I'm from the DTU, the Technical University of Denmark, and I will continue talking about viruses, but we will talk about quite different viruses. We'll talk about bacteriophages, but these bacteriophages are plasmid specific. So what that means is that they selectively infect bacteria that contain a plasmid. And so what really defines the host range of these phages is the plasmid content of the cells. So you see in this cartoon that if you have the same bacterium here and here that contains one or another plasmid, they might be infected by different plasmid-specific bacteriophages. And in contrast, the same plasmid-specific bacteriophage will infect two bacteria that have very different chromosomal background. So that comes in stark contrast with what I will call somatic phages, for which the host range is completely defined by the chromosomal content, and then as a result will infect only phylogenetically related bacteria. So how do these plasmid-specific bacteriophages work? The idea is that they typically recognize the conjugal pilus. So this is an appendage that is encoded by the conjugal plasmid, and that is really important to mediate the transfer from the donor bacterium to the recipient bacterium. So this specific recognition can be visualized using microscopy, as you see on the right-hand side of the screen. So why do I think these type of viruses are interesting? Well, two reasons. The first is that conjugal plasmid are very potent vehicles for antimicrobial resistance dissemination, and we've discussed that earlier this morning at the session. And the second reason is that I think these viruses can also tell us something about water quality. And I have a good reason to believe that, because it's actually already in use, specifically when we talk about the F plasmids. And if you paid really close attention to the presentation earlier in the session, so the work presented by Veolia, you saw that they reported some data about F-specific RNA phages as a water quality indicator. And this is a useful water quality indicator because the presence of these F-specific RNA phages indicate contamination by feces, and also because their fate in the environment resembles that of other RNA viruses. And so certainly enteroviruses, rotaviruses, and probably some of the other viruses that we've heard about today. So because this is a water quality indicator, we have an international standard that kind of guide us in how to measure these phages in the environment. And the way it's done, it's pretty low tech. It's this double Agar assay. So if you haven't seen this before, this is a picture. So the way it's done is that one mixes an environmental sample with a bacterium that contains the plasmid of interest, so in this case, the F plasmid that creates a loan of bacteria. And then we can see lyses, so plaques that are created and that really indicate the presence of plasmid-specific phages. So because this method has existed for a few decades, we have a pretty good data set about the environmental distribution of these phages. But I would like to point out that the F plasmid is only, well, a single plasmid and not necessarily representative of the other plasmid classes. So that's why in my group, we are currently trying to provide experimental tools so that we are able to detect, quantify, isolate plasmid-specific phages from the environment. And we want to do that for a few classes of conjugate plasmids that have relevance in terms of dissemination of antimicrobial resistance. Then we plan on using these tools to describe the distribution of these phages and essentially understand their ecology and what controls their fate in the environment. The way we've approached this is by being inspired by the double Agar layer assay. And what we've decided to use is a host that is a Salmonella enterica that has relatively low amount of somatic phages in wastewater. And we introduce one of 4 plasmids that belong to these 4 plasmids that are relevant in terms of medical importance. And then we tested the method using contrasting water samples that we got across Scandinavia. Notably, we included hospital wastewater, and there's a reason for that because we know that the hospital wastewater typically have a high load of plasmid compared to domestic wastewater. And so we thought that it would be interesting to check whether the phages would be more or less abundant in this sample. We use water of different qualities. So we use dead-end filtration for the low biomass samples. And then at the end of this work, we verified that we had assays that were pretty good in terms of repeatability and of limit of detection. So what did we learn using this? Well, this is a picture that shows the average abundance of these different plasmid-specific phages across different locations. So something you might be able to tell already is that river water really contain a very low amount of these phages, so we could barely detect them. And that came in contrast with our wastewater sample. So that really suggests that fecal samples are the main source of these phages. Their distribution was not simply controlled by the amount of biomass, so it's not something that is VSS driven, but we see differences so across plasmid classes and also differences across location. So it's not that hospital wastewater have always very large amount of plasmid-specific phages, but it's true for some classes. So certainly it's significantly different for the INCP class, whereas we see kind of the opposite for the INC-N class. So that's something that we can also illustrate here in this ordination plot. So again, you see that there seems to be a signature composition that allows us to distinguish the hospital wastewater sample from the domestic wastewater. The next thing we looked at is how do these phage behave in a wastewater engineering context. So we look at the removal in 3 Swedish wastewater treatment plants, and we sampled the influent in red, the clarifier, and the effluent, again, for the 4 plasmid classes. First thing we noticed was that by sampling 2 times during a week, that there was not so much day-to-day variation. The 2 data points tended to be very close to each other. what we saw is that primary clarification didn't decrease the amount of phage in our sample. So we see poor settling in the primary settler. But if we look then at what comes out of the plant, we could observe a very high removal. So typically there's 2 log removal across the plasmid classes. So we don't exactly know where the removal occur, but we suspect that might be associated with secondary clarification. So I come to my conclusion and perspective. So these data are really the very first one that describe plasmid-specific phages in the environment beyond the F class. We confirm that we find very high occurrence and abundance of this plasmid-specific phage in wastewater, so that seems to be the main source. The hospital waste did not contain an overall higher abundance of these plasmid-specific phages, but we could detect some compositional signature. So maybe there is a possibility to do some source tracking by using ratio of abundance across the classes we measure. We observed a high removal in wastewater treatment plants, and from what I could read from the literature, that's quite consistent with what others have also observed. Typically, secondary sludge tend to concentrate these RNA phages, but we don't have direct measurement of this. So we want to continue explore these phages and understand how they impact the dynamic of plasmid in the environment. So notably, can these phages replicate in the environment? It's still an open question. And if they do, something that we would very like to do would be to try to engineer the system and maybe try to use them as a tool to mitigate antimicrobial resistance crisis in the environment. So with this, I will thank my co-authors and the founding body, and thank you for staying all this time until the last presentation of the conference. I'll be very happy to take questions.
Congratulations, Arnaud, for this last presentation, indeed, and for having kept your motivation all through the conference. So, are there some questions in the chat? I have problems to see on my screen. It's so small. I cannot. Can you read the, please?
Yes, there's a question from Colleen. That's good. Not a lot was in the river water. Do you think there may be more in areas with less effective wastewater treatment and/or lower sanitation coverage? What was the type of wastewater treatment at the plants?
Okay, great questions. So yeah, about differences in different sanitation system with different sanitation system. I think that would be certainly true. And the river we sample was in Denmark, where we have a very good sanitation system. So I suspect that largely explained the result we've gotten. So I would be very, very interested in getting samples from other area and verify that indeed these phages can be detected in river sample that are somehow contaminated with fecal matter. And then the second question, yeah, the type of wastewater treatment at the plants. They were quite different. So the 3 plants had slightly different unit processes. Many of them had MBBRs. They all had activated sludge at some point, but at least no tertiary treatment. But yeah. So we covered, I mean, I realize 3 plants is not a lot, but we covered relatively different process design.
Yeah, in terms of phages, so we have seen at the beginning of the conference that you can make phage therapy to get rid of biofilms. Then phage seems to induce problems like antimicrobial resistance. They transfer antimicrobial resistance. And phage can be very useful to track of viruses, so as indirect measurement, so for monitoring of infiltration of water in environmental systems, in groundwater, across wastewater treatment plant. And phages manipulate communities as well, so they will impact, or there is this kill the winner concept that in enrichment cultures like Acinetobacter or whatever other organism, phages would statistically more affect the dominant organism. So how should we manage these phages now in water systems?
Big question. But yeah, for sure, I agree that it was super interesting to see in this conference how many people look at phages differently and with different tools. So I don't think we can give a single answer, but yeah, I certainly they have a potential for water monitoring, water quality monitoring, and certainly in places where there is no access to fancy DNA sequencing. I mean, as I showed in my presentation, the method we use, anybody with an autoclave can really do that, and you get the answer in 24 hours. So certainly there is potential for that. Should we be concerned about phages as a way to disseminate antimicrobial resistance? To me, I'm not convinced of that. So I'm following the literature, and to me, there is once in a while a detection of antimicrobial detected in phages particles, but I'm not sure how large a problem that is. So to me, I really think that plasmids as a horizontal gene transfer mechanism is a much bigger threat, because many of these plasmids are extremely contagious, if you like. So they are extremely good at transferring from bacterium to bacterium. And I think transduction is not as efficient. So that's why my idea is to use these phages as a tool to get rid of the bacteria that contain plasmid selectively. And yes, that will create DNA floating in the system that may be taken up. But again, I think the rates at which this happen would be beneficial. So I'd rather get rid of plasmids in live cells and have naked DNA floating. But that's, I mean, we certainly need more numbers.
Thank you very much, Arnaud. And I suggest that we move for some minutes for some global discussion. We stay in the same room, and progressively we will close the conference and we will finish on time. So it's important to always have buffer time in a conference. So you start at a certain point, you finish on time. So we have seen with Colleen the COVID-19 dashboard, with Diana affordable protocols for SARS-CoV-2 concentration and measurement, with Xuan how we can look at these genomic variants beyond the concentration of these SARS-CoV-2 viruses with multiplex PCR sequencing, with Pauline what are the quality standards that are needed for reclaimed water for SARS-CoV-2 but other pathogen agents, and with Franciscus about other agents and other viruses of importance that need to be tracked. He also pointed out the urine, so that some viruses seems to shed in urine. We'll maybe ask Colleen then, should we do COVID urine 19 dashboard? COVID urine 19 dashboard or virus urine dashboard instead of only the poo. And with Arnaud, we saw that phages are also water quality criteria markers and can be useful to track other viruses at the same time. So maybe to start with this basic question, if I come back to my story of this discussion with Emmanuel Adler from École Centrale de Paris, looking at poo, looking at urine. So the doctors were doing like that in the past, and we are still doing like that in medical measurement. We give all samples of urine and poo for measurements, and here it goes in the same direction at wastewater level or water level. So what about poo versus urine? Should we track everything? And when poo is important, when urine is important? Somebody has an idea. I don't know if Colleen, as keynote lecturer, you want to start to answer this
question? Yeah, sure. Great point. Yeah, well, I'm always open to another themed poop or urine account. For COVID poops, we do mean to incorporate urine as well, or like in the wastewater sampling for SARS-CoV-2, it is all combined together. I mean, some are sampling the influent and some are the solids, but things are settling out. But mostly SARS-CoV-2 is shed through the feces. I think it's only about like when they're doing hospital like samples of cases, it's like only like 2% shedding in urine and like mostly in feces. But definitely we should be looking at the full mix. But that was interesting to hear that about different viruses and how some are shed more in urine versus feces.
And the wastewater and sewage is the full mix actually. So how can we combine targeted links to poo, to urine, to wastewater? So what can maybe wastewater in the future help to further look at the health state of populations in a city? What kind of markers could be interesting to follow beyond viruses, but to look at disease emergence in a city?
Right. Yeah, I don't want to steal all the question answering. I know there's a lot of great speakers on here that can answer from my perspective. I mean, there's also urine separation technology, but that's not necessarily like ubiquitous everywhere where you could like urine diverting toilets where you could sample a little separate, though there is some cross contamination. I think for cities, yeah, I mean, Arizona, other places we're using like wastewater to monitor the health of a city and other biomarkers. It was used for opioids or illicit drugs. There's also personal care products and you can monitor stress and everything. It does get into the ethics of wastewater and people feeling like, oh, you're taking that information from my like feces or my waste that is like considered private. So we do need to be like careful of how we approach that and like have a kind of a community engaged approach that we're doing this for the health and we're not prosecuting people or taking like disciplinary action, but I'd welcome others to respond too.
Maybe Franciscus, since you brought this point on urine, so what is your viewpoint here?
For my viewpoint, in terms of tracking targets in wastewater, I completely agree with what Colleen says. You know, your usual virus, bacteria, biomarkers are, in a sense, fair game. But when you start to touch on, you know, other targets, maybe like human genes or something like that, that like treads closely to privacy issues, which I think the wastewater community needs to kind of like be in contact with, you know, public officials, with other you know, bioethics to properly manage this. Because yeah, if it's like bacteria and viruses, it's like quote unquote public, but when you touch on these more sensitive targets are quite sensitive. Yeah.
Thanks. And Colleen mentioned urine separation. I remember, for instance, colleagues at EAVAG worked a lot on the Reinvent the Toilet Challenge. So what type of measurement should be done to verify that, for instance, products recovered from urine do not contain potential problematic viruses? Or what kinds of treatments should be implemented to get rid of the viruses? Maybe, I don't know if there are some people from UBC as well. I know that they also work on, for instance, production of struvite from urine separated at the source. Are there some regulations to look at viruses in these recovered products? I don't know if you, Colleen, you have any idea about this.
Yeah, I mean, like back in when I was in Peace Corps in Mali, we would separate urine and like we'll let it set for a day and then apply it to crops as like a fertilizer and dilute it. And like mostly that was thought to have like removed all the viruses just because of, you know, the pH and but then schistosomiasis could still be an issue or transmission. So I think there's ample like space there to research more about extracting data from that or monitoring pathogens. And especially, I know there's a lot of research in like Vermont and Arizona and around the world, and I'm sure with the Dutch as well, for pharmaceuticals and other products in urine that are like very persistent.
Thanks, and maybe let's move to Xuan and Diana. So Diana brought the point that we need affordable protocols, so low-cost protocols for implementation, notably in low and middle-income countries. To which extent we can apply genomic technologies then? So how can we make not only the protocols for concentrating the virus cheap, but also the genomic sequencing then cheap? So Xuan, would you have any idea, or maybe Ryan as well from UBC, would you have any idea how can we make sequencing cheap and affordable, and maybe with the nanopore or the portable sequencing devices, is there a way that we can bring the sequencer in specific location and sequence these viruses directly, but at low cost? Xuan, are you still here?
I can. This is a good point, actually. Yeah. Could you hear me clearly?
Yes, sure.
Yeah. So this is actually a good point. One is the cost of the sequencing. So if we're sequencing on a Illumina sequencer, that's say a more traditional way, second generation sequencing, it would potentially costing more per sample, and you will need a large amount, like collecting a bunch of samples to be sequenced together, which is not really a real-time monitoring method, ideal real-time monitoring method if you don't have that many of samples to sequence. So nanopore, as you bring up, nanopore sequencing is actually a really good choice for on-site monitoring, and people have more.
Your line is bumping, sorry. Can you repeat your last two sentences?
Virus on site with sequencing cost.
Xuan, there was a problem. The two last sentences that you mentioned, can you repeat them, please? Okay. So, and Diana, are you still here? So, how do you see on your side not only the concentration of virus, but the measurement after the concentration? So, how can we make this measurement affordable and easily implementable? And the data processing as well, probably afterward, needs also quite some resources.
Yeah, so what we think is that at least if you have a method that it's simple to do in a small laboratory, then you can send a sample and the sample can be processed in a molecular biology lab. And so that was our idea, because I don't know if there is a way to get simpler or cheaper methods for detection. So I think that it's maybe more difficult to get. But at least this way, so if you concentrate the samples and then you send them to a lab, and this lab is doing that already for clinical samples. So we think that this way, at least you can get some information.
Thank you. Now I have a creative idea that just popped now. Based on the dashboard, we can identify location based on geoinformation systems, so hotspots that need maybe more measurements, more regular measurements, etc. We discussed at the very beginning of the conference about autonomous systems for sampling and monitoring. Would there be a way at a certain point maybe to equip drones with some sequencers that can be deployed in areas of concerns if we cannot go physically in these areas? So if we think about all other types of viruses, other types of pathogens that are really critical, I don't know, for instance, like Ebola or other types of disease, do you think that we could deploy low-cost, affordable technologies that are autonomous and that you can bring them with drones or other types of device where you don't need the human intervention on the analysis? It's a bit creative, but maybe something is already in place for that, for technologies for sequencing samples from Mars, or I don't know. Somebody has an idea about that, or maybe Amit, since you work on this autonomous system.
So I'll make a comment there. It's not on the sequencing side of things. I think there is already something something, where is my camera? There, something is, stuff like that is happening on the imaging side of things, trying to image harmful algal blooms in pretty remote locations where, you can get data from satellites, but the satellite data is not always have the resolution that's needed. So there are groups that are using drones to kind of image that. I've also seen examples of where drones are being used to. So this is a fun example where they were. I don't know, what is it that whales do where they blow out? What is it called?
Jet. What a jet?
Jet, exactly. So I'm pretty sure I saw a paper where they had a drone flying over to trap that sample to capture the microbiome of a whale snot. So I think people are thinking creatively. I think so certainly these are possibilities. Yeah.
I think one of the first aim of this portable sequencer was indeed, if you cannot transport the sample from one location to another location, and besides the disease, there are also all the Nagoya protocols versus biological samples, so also for non-disease samples. So you cannot always transport samples, but you can transport the sequencer, or you can transport an infrastructure that can sample at the same time. Yeah, so that can be interesting. Yeah, so progressively we should gently close this discussion and conference. So it was a pleasure to be with Francesc and Sergio and Mark at this table today, with all the persons in the room and with all online. So I think we are very impressed of the quality of the presentations from everybody, from keynote lecture to oral presentation to poster presentation and roundtable, and you maintain this high level all through the conference over 3 days with 3 sessions per day. So congratulations everybody for your assiduity and dedicacy to this conference. Thank you so much. I think we can say a round of applause to everybody. And I will just move to the console for some acknowledgement and some final words. I love this. So let's continue. I go to the next part. I'm back. So Eric, we are here. And if you want just to straighten your legs, your arms, just stand up, and you can take 2 minutes. I just put the final slide set that I have. It's not so long.
I think there was a bit of miscommunication. We moved. No, que estoy hablando por mi ordenador. And we moved to the next session in day 3 closer in the program. So you can join the call there, where David will talk now.
Great, thanks.